Thank you. Thank you very much for that kind introduction. Thank you, Venerable Aoife, and thank you, Naomi, for putting together such a good meeting. I'm going to do something different than what has been done so far. I'm going to not talk about a single cell. Well, I'm going to talk about cells, but not at the developmental stage. We're going to talk about cells in adults. And we're going to talk about engineering those cells for therapeutic gains in adults with specific diseases. So there's something very different from what we have discussed so far. The connect to ethics and Buddhism are not directly apparent, but hopefully we can have that over questions and discussion as to how I think this has bearing towards how we look at an individual and how we understand pain and suffering in an individual. But first, I'm just going to take you through the science that we do. So bear with me for the next 15-20 minutes. It'll be a slightly more science-heavy talk. What we look at primarily are individuals with type 2 diabetes who tend to receive various types of synthetic materials as a means to cure a particular aspect of type 2 diabetes, and I'm going to take you through two aspects. We look at how materials that go on to individual with diabetes, how the immune cells respond to that, and in the context of two different diseases, lung infections, and I'll give you some context, and diabetic foot ulcers. And so there are these two different ideas that I'm going to introduce and some data on each of these and some insights, not from human studies, some human studies, of course, that we have been doing, but mostly from animal models of research and how that has helped us understand how humans might be suffering from similar diseases, but also raises questions on ethics on how we can use animal models of research for the same questions. When we start looking at lung infections, I'll begin with that. I think the first thing that comes to mind is this. There was this first report that came out very early in 2020, September 2020, when the rest of the world was still sort of grappling with the first wave of the COVID pandemic. Italy had gone through a serious problem, and they came up with this study showing that people with type 2 diabetes tend to have greater mortality when infected with COVID. This was expected by scientists, but was not necessarily something that the policymakers had planned for. It was not clear who would be the ones who would be most affected by COVID-19. And this was the first study that showed, yes, it is most likely going to be individuals who already have some sort of disease, most likely obesity and diabetes. But if you go back, why did scientists think this was going to happen? In the 1990s, actually 1970s to 80s, and then the final study in 1990, showed that individuals with diabetes needed influenza vaccination. And in the US and in the UK, this led to the program where everybody started getting influenza vaccines every year, which is still not practiced in the rest of the world. In certain countries, yes, but not in every country. And the reason for this was there's much higher chance of influenza causing disease and death in diabetes individuals and less in other healthy individuals. And the exact reasons for this are still unknown. Even though we knew this about 35 to 40 years ago, we know this from COVID-19 now 5-6 years ago, the exact reasons of why this happens still remain unknown. And that's something that we were interested in studying. We actually started, even before COVID-19, looking at this paper, but it sort of helped us that you understand this better in the context of COVID-19, so I gave you that example. All right? So what exactly happens? Well, type 2 diabetes now is thought to be an inflammatory disease. And what I mean by that is, apart from having high blood sugar, which causes various issues, you end up having immune cells that don't function as they normally should, which is to destroy invading pathogens. And that's sort of the straightforward function of immune cells in humans. And they tend to start destroying your own self. And that's what we mean by an inflammatory disease, where they start attacking your own body in various forms. And if I were to depict it in a form of a cartoon, this is what happens. You have stem cells. We talked a little bit about those stem cells in developmental biology. In normal conditions, they're producing your blood immune cells that are in circulation that get to the tissues upon activation if you have an injury or an infection. if you have something like diabetes, you tend to produce more of these cells for reasons that remain unknown, but they have more numbers in circulation, and they tend to get secreted in larger numbers at the tissue site if you receive an injury or activation. But what happens if you have multiple such injuries? That is the example of if you have diabetes and then influenza infection, or if you have diabetes and then COVID-19 infection. What ends up happening? That still is unclear. Do we get more cells in blood? Do we get more cells in tissue? These sort of questions are tough to answer, especially in humans, because these sort of studies have not yet been done. And so we decided to try to ask the same question in an animal model of research to understand better what type of treatments might help those individuals, right? So the way we look at it, if you have chronic inflammation, let's say type 2 diabetes, your life is a new normal life. You can survive, you probably take some medications, you eat less sugar, you maybe exercise more, but you have some sort of new normal life. That's how we look at it. But now, you suddenly have an inflammatory event, maybe an infection or some injury, and suddenly that normal life changes and you have increased mortality. And so you basically have gone down a different path because of that singular event, which has been, and there are many other events before that that triggered this event. So our question is, can we reverse this, or can we try to bring it back to this new normal life? What are the reasons to this increased mortality? Right? And so we looked at it in diabetic mice and in humans, and this is just some data. I don't mean to belabor the point with these sort of graphs, but basically I wanted to show you that one particular cell type, the neutrophils, are dramatically altered in individuals and mice with diabetes. They tend to be much higher in diabetic individuals compared to healthy controls. They're functionally altered, so basically showing for the first time that yes, there are these cells that are different, right? So what do these different cells do? Well, now what we are trying to say is we can replicate this whole condition in a mouse model. We have a chronic inflammation, diabetes in the mouse model, and then we give it an inflammatory injury. So we induce what we want to, which is sort of similar to an infection, but we don't give an infection, so that we can model and understand how these cells change. And basically we assess lung health, just like in humans, we assess them 4 weeks later. And this is a sort of graphs, pictures that you get about the lung itself. These are known as histology slides, and the lung is supposed to have cavities where you can have air pockets. And in the diabetic mouse, and very similar to diabetic humans, you tend to not have those air pockets. And so people tend to not be able to breathe. And that's one of the reasons that you have increased mortality, right? And that's because that can be counted as an injury score. So in diabetics, once you have some sort of infection, you have greater injury, which leads to less amount of air pocket, allowing less amount of air to be taken in, okay? So what we have shown is that diabetes, and there's lung injury, but there are many different cell types. And this is where we'll use a traditional reductionist approach that people use in science. In diabetes, you have 20 different cell types that are changing. How do you know that one cell type causes the other, causes a particular event, right? Like lung injury. And in this sort of reductionist approach, we think of it very simply as the following. If you want to say that an entity X, in this case are neutrophils, a particular cell type, causes a condition Y, which is higher lung injury in diabetes, then we could either delete X, which should prevent Y, if that was the case, or increase or add X to increase Y, right? So this is a very reductionist scientific approach of removing something that causes a disease or adding it to cause the disease. And this is what we tried to do in that particular model. And what we did was, this is where the engineering part comes in. It's very difficult to completely change a cell type in humans and in animals. But we are able to do this using biomaterials that are commonly used for cell transplantation. And that's another area of ethics that I'm not going to get into. But you can encapsulate cells into these sort of microspheres. They are protected then from the body, and those microspheres can be implanted at various sites in the body. We do this regularly for type 1 diabetic animal models, and we are working towards trying to develop this for type 1 diabetic patients. Nevertheless, you can use the same biomaterial implant, place it in the abdominal cavity where there's a lot of space, and create what we call an inflammatory event that is similar to diabetes in that it changes the neutrophils. And that's what this particular student in my lab did. He showed that almost all the changes that I showed to you earlier in diabetes occur when we implant biomaterials, because the body sees these foreign substances, the biomaterial, as an inflammatory agent, similar to what happens in high sugar conditions in diabetes, or similar to what happens in any other disease. So this is something new, but it is also well known that anything foreign is going to cause these sort of reactions in the body, right? Then we use this new technique called single cell RNA sequencing, and the idea behind it is that we can take individual cells and understand the fate of these individual cells. And here's where it becomes a blurred line on, are we understanding the fate of individual cells as an individual entity? Are we understanding the fate of individual cells in the combined entity that is the organism? But here, from a reductionist approach, we think of it as the fate of individual cells as individual entities, which then affect the entire organism. And that's a key distinction that we make here. And so when we look at it, we can look at different cell types, and each of these dots here are actually a single cell. So we're looking at millions of cells together that are present in the body. And you can then characterize them as to what is happening. And we show that no other cell type is dramatically changing except for the neutrophils and the monocytes, which are related cells, which goes to show that we're only affecting those two cells with our biomaterial implant model. And that gives us this sort of hold onto this model to show that, okay, nothing else is changing. If these are the only cells that are changing, can we do something about it? Can we show that these cells contribute to a particular disease? And that's what we end up doing. So this is the summary so far. We have a biomaterial implant that results in a large number of these cells. Could this be used to understand why there's increased immunopathology in lung infections and so on? So we do the same model after the biomaterial implant. We assess the lungs after giving an injury. This is just a model to show what happens. And you can look at the lung damage the same way. In the earlier, I'd shown you some pictures. I'm not showing you those pictures again. But basically, this is the biomaterial that we use. And this biomaterial causes the same type of injury in the lung. Even though the biomaterial is far away, it's in the abdominal cavity. But it is causing a systemic change, causing lung injury, when we give the animal infection or some sort of agent in the lung, right? And we can now break this down even further. So again, a reductionist approach as to what individual aspects of the lung are changing. If you ask a doctor, they'll say, oh, the septal thickening in the lung will reduce airflow. Hyaline membrane is important for compression and expansion in the lung. Then proteinaceous debris is a sign of inflammation. Each of these are aspects of the lung that a doctor studies and says this is going to get worse or better, right? And we show that the same thing happens in individuals that have been implanted with materials. And so this is a sort of summary. This is where we are at right now. In the bone marrow, you have these stem cells that are producing your final immune cells, the neutrophils. Those neutrophils are getting into the blood circulation. If you now give it an injury or an inflammation, which is type 2 diabetes or a biomaterial implant, we can increase a particular type of cell known as the immature neutrophils. This happens in humans and in animal models. It's a single change that occurs, and that single change in the presence of a second inflammatory stimulus like COVID-19 or influenza causes more lung damage. So basically we are saying that this particular cell change is driving the damage in the lungs of that individual. And what we've been doing now is to actually develop new therapies that can block this, and this could potentially prevent increased mortality in individuals who end up having infection, especially diabetic individuals that have an infection. None of these were used in the COVID-19 pandemic, but now we think that individuals who get COVID-19 or various other strains of COVID or influenza infections could benefit from these sort of therapies, and that's what we are taking forward to clinical trials now. So this shows how we can use a biomaterial implant to understand the human disease and then potentially intervene using reductionist approaches, of course, which brings into question a lot of different things about the how we define a cell inside our body as an independent entity or as part of a whole. So that's with respect to the lung infection. Can we do the same with respect to something else? in the same diabetic individual, there's a different model or a different disease that comes up. These are foot ulcers. I don't know how many of you know of this, but about 10% of diabetics across the world will develop an ulcer in the foot which doesn't heal. This is because the healing is compromised in most type 2 diabetic individuals. Here's where I want to take a small step back and an anecdotal story. My lab, and I had been working on it. We had heard about it. We had read it in papers. But I'm not a clinician, so I didn't see it in real life. And surprisingly, at one of the oddest places, according to me, I saw it in real life, at least in India. This is when my grandfather passed away, and we took him for cremation, took his body for cremation. It was a sad time, but he had lived his life in some ways. He was at the very end of his life. He was suffering. He was 89, and we felt like it was fine. We had him for a long time. It was a good death in some ways, as we call it, right? In my mind, so I was just looking around, and this is the first time I'd been to that part of the country where my grandfather grew up. This was right next to the Ganges. And I'd actually never been to the Ganges other than that. So this was the first time also to the Ganges for me. I'm just looking around, and there's a line of bodies that are being cremated. And this is a very, I don't know if that's the right word, but popular cremation site, if that goes. And I notice that if the individual was older or than a certain age, about a fourth of them had an amputation. One of the limbs were missing. That's interesting because most of those amputations are because of diabetes. If they've lived long enough, yes, there are individuals who have amputations for other reasons and misfortune of other causes. But in many cases in India, if you have a lower limb amputation, especially at the ankle or at the toes, it's because of diabetic foot ulcers. So till I saw that image, I didn't realize how many people have this issue. And interestingly, it's also among individuals who tend to all have some money that they are able to afford a good life for themselves, but at the same time end up also eating food that is not necessarily as good for them, and eventually end up getting diabetes. And this is a social phenomenon in India. May not be true elsewhere in the world, but this is what ended up happening. And that sort of brought to sort of sense of why we're doing this in our lab. And the main goal is that in India, it's not necessarily true that you have to be well off to get diabetic foot ulcers. In fact, most of the people who get these amputations tend to be people who are not very well off, but just about well off enough that they have good amount of nutrition, and not very well off in that they don't have education to treat that disease quickly enough, and eventually end up getting amputated. And so we're trying to address that phenomena, and we realize that more than 95% of the individuals don't have a particular treatment right now. The treatment's either too expensive, or they don't work, or they're not easily accessible for 95% of the Indian population. So in that context, we were trying to develop something that is completely different from what is available and as less expensive as possible. So let's get to that. Why do these diabetic foot ulcers not heal from that anecdote, and what can we do about it, right? So we performed a clinical study after that in Bangalore, and interestingly, there are not too many clinical studies in India that are conducted on Indian patients. We tend to sort of borrow data from the Western populations and tend to say the same thing would apply, but that's not necessarily true. So we recruited 80 individuals who had early stage foot ulcers. We followed them up. This is done at a clinical site, and we provided them with the best state-of-the-art clinical care. In addition, we performed this sort of flow cytometry experiment where we look at their immune cells, again coming back to that immune connect, because immune cells are altered in these individuals. And we get this sort of data. And this is where the AI comes in. There's some more engineering that comes in. If you want to write down this data, we end up having this sort of form, right? And I don't mean to sort of get you to read this, but just to say, we have data now on 52 individuals and 45 such columns, and these are numbers. These numbers lend to very simple statistical analysis that was developed 70, 80 years ago in a formal sense, but probably has been used in various forms for the last millennia or so, which we now call machine learning and AI. It's basically crunching these numbers in a way that we can make sense of them. And that's what we did, and you can reduce this down to these sort of key variables using a method called logistic regression, which is simple statistics. And it comes down to these 4 parameters, 3 of which you would get in any hospital that is across the world if you go for a checkup. I believe most of you would have gotten a cholesterol checkup done. I believe that most of you would have gotten also an LDL cholesterol level checked up. If you're above 50, you should definitely get these 2 checked at least once in your lifetime to prevent other diseases from developing. This is the number of monocytes. This is sort of the first check that a doctor asks you to get what is known as a complete blood count. How many monocytes and neutrophils do you have to understand if you have an infection versus some other disease. So these 3 are common tests done in any individual, and we are saying that individually, these tests do not mean anything, but in combination now with this statistical learning model, we can predict whose ulcers will not heal. And that's a big step forward because these patients come to the doctor and they may come once and they may never go back. We need to be able to tell them that your ulcers are not going to heal when they come, that one time, that's it, you get one chance. And so if you can just do these 3 tests along with the small additional test that we add, we feel that we have 87% chance of getting accurate results on whether their ulcers are going to heal or not. And that's a unique feature that we provide to these patients. And now we're developing this into a clinical trial that as a diagnostic marker, saying that all we need is these 4 markers when an individual comes to us, and these can be done within 3 hours in most hospital laboratories. And together, we can tell if this individual's diabetic foot ulcer is going to heal or not, which is a big step forward because now these patients who are not going to come back to the hospital, if you tell them there's an 87% chance that their ulcers are not going to heal, they may come back to the hospital to get their foot looked at more often, which really prevents that amputation, right? So to take this forward, that's not the only thing that we need to look at. Okay, we tell them that your ulcer is not going to heal, but if you tell them I can't do anything about it, there's no point. And the treatments don't work currently. In about 65% of the individuals, the early stage foot ulcers, it works. 35% it doesn't work. And once you get to late stage, it is like 80% it doesn't work, 20% it works. So it's really poor treatments right now. So if you look at the wound healing cascade in humans and any mammals, these are the 4 steps that happen. So immediately after a cut, you have bleeding, and that leads to clot formation. Then there's an inflammatory phase, which is the redness that you see and the swelling for the next 3 days. Eventually, you will have a proliferative phase where you have scab formation, regrowth of tissue, and eventually your wound heals, right? And that's the complete healing process where you have remodeling, scar formation, whatever else. Now, what ends up happening in diabetic individuals is you're stuck at this inflammatory phase. You don't move to this proliferative phase. So what you end up having is continuous redness, no closure of wound, so it's an open wound forever. And that's the stage we were trying to address. We decided to use a therapy that is different from what has been done before. We used an immunomodulatory drug that was used in the early 70s, that was discovered in Japan in the early 70s as a immunosuppressant for transplantation, but has since been discarded because it has kidney injury issues. But if you can apply it locally at a foot site, you won't have those kidney injury issues. So we use that drug. We combine it with a biopolymer that can go on as a bandage, as a cheap biopolymer, not the expensive ones that cost over $1,000, but we've been developing ones that cost less than $10 with the same purity. And we combine these, sorry, there's just a little bit of data there, to show that we can have accelerated wound closure. And this is the bioengineering approach that we're talking about to help heal those wounds. This is what that scaffold looks like. This is a ruler, so it shows that it's about 2 centimeters. So if you think about it on your toe, it can easily fit in, and all I have to do is then attach gauze. This is what it looks like when you look at it very closely. It's a simple material that swells, so it'll soak up any of the fluid that leaches out of your body. That's a common occurrence, so we wanted to design that. And basically, we show in animal models that with this drug, this green line here, you get much faster wound healing compared to normally. So basically what we're trying to say is that in the animal models, we're able to show that these drug-releasing chitosan-based material scaffolds have a low cost and easy to develop bandage that cause healing, and we are hoping to now translate this into humans as a way to provide faster healing in diabetic foot ulcer patients. I'll stop here with these concluding statements. Our goal has been to use materials that are less expensive, but many materials are being used in clinical practice already. And so if you go to a hospital right now, the chance that you'll have a material injected inside you or some sort of material placed on you is very, very high. So you're starting to come into that interface as an adult of what is biological and what is abiological or material. And they are essential for treating many ailments. Most of these are very expensive, and our goal is to try to get them to a less expensive one that's affordable to the 90% of the world population. Right now, most of these material implants are affordable less than 10% of the world population. But a key question remains, for us at least, do we focus on developing new therapies that would be expensive? Do we focus on repurposing older therapies? Do we focus on developing new paradigms for therapies? And that's a question that will still need to be answered in this question-answer session. Thank you very much for your time. I'm very happy to be here again. Thanks.